Everyone, am I audible? Please confirm if my screen is visible and I'm audible to you all. So, today's session, it's on Unit 5, Subunit B. We had discussed early development in some organisms, C. urchin and C. elegans, but today we'll be seeing some more in amphibians and in Drosophila, in cheek. Okay, all this we will see, and some C. urchin series also. Yes. Anyone there? Please confirm if I'm audible. Shall we get started now? All right. The first question is from June 2021. This is regarding an experiment which was done on Xenopus embryo. Xenopus embryo means amphibians, and there are some activin secreting beads. Now you have to know what is this activin, what is the function. They were placed on unspecified cells from an early Xenopus embryo. Early means the development it's not completed. Still some signaling can be taking place, and they were placed in unspecified cell so that they can get the specification. The activin then diffused from the beads. Secreting beads means those will be artificial beads from which activin will be secreted. Now the beads contained 1 nanomolar of activin. Beads means you draw like this, a rough diagram. Always the approach for such experimental question is drawing a rough diagram. Okay. So if you have the beads, then from here slowly and slowly they will be releasing the contents, right? So if the beads contained 1 nanomolar activin, it elicited, means it promoted the expression or increased the expression of XBRA gene in the cells near to the beads. So whichever is near to the beads, okay, they will be getting 1 nanomolar, and in that the expression will be elicited. But if the expression contained, if the beads contained 4 nanomolar activin, you increase the concentration, the expression of XBRA was elicited in cells, but only when the cells are at a distance, not close to the bead. Okay, so basically what do we get to know from here? That 1 nanomolar activin and 4 nanomolar, one is lesser. When it is lesser, then the XBRA gene activation was near the beads. That means at a low concentration of activin. But when you had increased the concentration, obviously from the beads, higher concentration will be released. But in those cells, XBRA was not elicited. Okay, but it was done only at a few distance away from it. Beads with no okay. In the later case, expression of goosecoid was observed near the source bead. In the later case means where you had more concentration. Near the beads, XBRA couldn't be elicited, but the expression of goosecoid was there. Okay, so what can we draw a rough diagram? That here is 4 nanomolar. Okay, so near this the cells, no XBRA. Okay, XBRA, no XBRA you can write, but goosecoid is there. This is how you draw rough diagram. Goosecoid is present. Okay, and surrounding cells, XBRA is there, right? Beads with no activin did not elicit the expression of either of the two genes. So if you take nothing in the beads, then no XBRA, no goosecoid, either away or closer to the bead. And the first case was where you took 1 nanomolar. So near to the beads, you saw XBRA, but no goosecoid. Okay. So what is your conclusion from here? Clearly we understand that you have low concentration of activin. If there is lower concentration of activin, then XBRA will be high. Okay, but question is what is XBRA involved in? So that is what you have to remember. Which of the above statements are correct? A only, C only, A and C, B and C. So, any guesses? Yes. Whoever is attending, any guesses? See, there is no harm in making a mistake. Anyways, I'll correct it. Try to guess or try to, not guessing obviously, try to solve this question and give me whatever answer you think is appropriate. Okay, Sarita says option 3, which means A and C both. Okay. So concentrations of activin that do not lead to expression of the two genes specify the cells to become blood vessels and heart. Which side is blood vessel and heart? Blood vessels and heart will be from the ventral side, isn't it? This is a question regarding this axis formation. And the dorsal side, see dorsal most sides, those are where the notochord and nervous system will be triggered, neural tube. Okay. So you should know that XBRA, it is triggered where it is triggered, that part you should be knowing. So let's see if Sarita's answer is right. So yes, you're right, Sarita. Now, these are the diagrams which I mentioned to you. See, this is 1 nanomolar. This is activin, blue color is activin. We drew rough diagram, but these are the actual diagrams of the experiment conducted. So this question has been directly taken from an experiment. Okay. And you can see near the cells, obviously, if the concentration is low, the diffusion can happen only nearby, isn't it? So that is why it is happening nearby here, as you can see. Okay. And XBRA, you can see this activin bead, if we take more concentration, then see where is XBRA expressed? Not in the yellow region, because here the concentration of activin will be more. This is what is the 4 nanomolar. So the concentration will be more outside. Okay. And no activin means if you remove the post removal, then also they stay some same like that. But if you don't take any bit, then neither of the genes are getting expressed. Okay. So since 1 nanomolar is low concentration, less activin was diffused, and also in 4 nanomolar, the distant cells would get less activin and not the nearby cells. Okay, which received high activin concentration. So wherever goosecoid is there, that is nearby to the high concentration 4 nanomolar, there we did not see XBRA. That means what is our conclusion? That goosecoid inhibits XBRA. And wherever XBRA was there, goosecoid was not synthesized. Goosecoid was only synthesized when XBRA was not there. That means XBRA also inhibits goosecoid. Okay. Hence, it can be concluded that in Xenopus embryo, activin induces a range of mesodermal cells in a concentration dependent manner. The genes XBRA and goosecoid are activated at low and high respectively. That means XBRA will be activated by activin at low concentration, and goosecoid will be activated by activin at high substrate concentration. That means high activin concentration. Now I mentioned to you, you should be clear of what does the goosecoid and XBRA do. So goosecoid, it is very much involved in the dorsal specification. This is a homeobox gene. It is involved in dorsal ventral patterning of the mesoderm, and it is expressed dorsal lip of the mesodermal tissue. So if you have low concentration of activin, it will not express the goosecoid. And if it cannot express goosecoid activin, then it cannot lead to formation of dorsal most structure. So low concentration of activin means the goosecoid will not be synthesized, and in that case there is no dorsal structure. So since here B option says lower concentration of activin helps specify the dorsal most structure, that is why this is wrong. Anyways, we are understood this, that high concentration of activin activates goosecoid, whereas lowest concentration activates XBRA. So that was A and B. Now coming to option C, when animal caps are treated with this much of activin, they differentiate into ventral mesoderm. So this is lower concentration where XBRA gene will be triggered, but not goosecoid. So which includes in that case at lower concentration when XBRA is there, So that will trigger the formation of ventral mesoderm, which includes mesenchyme, hematopoietic cells. So that we got that blood and muscle cells, what we got. So that will get triggered by ventral mesoderm. So if activin concentration is so less that neither of the genes are expressed, then in that case they will not be able to form hematopoietic and mesenchyme. In that case they will be forming the blood cell and the mesenchyme. That is, it is even less than 1. Got it? So next question, it is based on, I hope the other students are following because I'm not receiving any response from you. Please actively participate. Given below are few statements regarding the role disheveled and beta catenin play in the development of sea urchin. So this is regarding sea urchin. Here, this is a question on opposing statements. One says DSH is localized in vegetal cortex, another says DSH is localized in cytosol of the oocyte. Then beta catenin accumulates predominantly in the micromeres and somewhat in the upper category of cells, also upper layer of cells, which is the Veg2 layer. And if you treatment of embryos with lithium chloride does not allow the accumulation of beta cat in the nuclei of all blastula cells. Okay. And beta cat, when it is prevented from entering nucleus, the embryo becomes a ciliated ectoderm. So you would be knowing that in sea urchin, you have these layers of cell. The bottommost layer, those are the micromeres. Specifically, you have the small micromere and then the large micromere. And then you have the wedge cells. So the bottom layer is wedge 2 layer and then the top layer is wedge 1 layer. And up you have animal cells. So animal 2 tier and animal 1 tier. Okay, AN1 and AN2. And the large micromeres, they get specified autonomously. They get specified into the skeletogenic mesenchymal cells, right? So those will give the signal to the cells lying above them to form the wedge 2 layer. They will in turn give the signals to the cells to form wedge 1 layer. So basically this autonomous specification of the large micromere, it comes because of beta catenin. What is beta catenin? It's a transcription factor. Okay. So here, which one of the following options represents a combination of correct statements? So these are your option statements. So script is not clear. How to understand? Okay, is everyone else facing the problem? Is the script not clear? The question is not visible to you, is it? I understand Chettiar Enterprise, but in order to solve the question, the students need to see it completely, full screen. Okay, that means the entire question in one screen. So that's why these questions are really very big. That is why they have to be a little bit smaller, probably. But then if I zoom it, or you will only be able to see only one part of it. Okay. So I hope it is fine. Can others confirm? So, anyways, I'm reading out the question to you also. Okay, Sarita says it's fine. All right. So, what would be the correct answer combination here, everyone? Maybe check your enterprise. You can try to make it full screen, like the YouTube, wherever you're watching. Down there will be a full screen. You can click on it. Then the questions will be more bigger. Font will be more bigger for you if you're watching in mobile. Okay. Anyways, others are able to see, so probably full screen will help you. All right. C and E, but you have to select three options. Are you not sure A and B which is correct, Sarita? See, anyways, the vegetal cortex is which one? Whichever side the nucleus is there, that is the animal side, and the other side is the vegetal side, isn't it? And from there only we get this veg 1, veg 2 names, right? So, since the large micromeres are towards the vegetal side, this is where the large micromeres are. So that means your beta catenin, they are the autonomous specification factors because of which large micromeres they will get specified. So they have to be in the vegetal section. Okay. Now, if you don't know the correlation, see unit 5 and unit 4, they have a very close relation with, unit 5 has a close relation with unit 4, that is cell signaling. Okay. No, Sarita, they will be present in the vegetal cortex. So that is why A will be correct. Okay. Here A, C and E will be right answer. Yes, option 3 will be correct. Correct. Okay. So see, role of beta catenin in specifying the vegetal cells. Micromeres undergo autonomous specification to become skeletogenic mesenchyme, and these micromeres will produce the initial signals that specify the other tier of cells. Okay. So here you have this entire signaling process. We have the frizzled receptor. This is WNT signaling. You should know about it. WNT receptor, when it binds to this frizzled protein, they will get activated, and in turn they will activate another protein called as disheveled. Okay. Now disheveled will be inhibiting a protein called as um, this complex, not a single protein but this entire complex, which is GSK APC axin kinase. Generally, in a cell where there is no WNT signaling, this complex will be formed. But if there is WNT signaling, then this complex formation will be inhibited. Now, if the complex formation is there, then this kinase, GSK is a kinase, it will phosphorylate a protein called as beta catenin. And because of phosphorylation, this ubiquitination will be done, and beta catenin will be degraded in that case if there is no WNT signaling. Okay. But if there is WNT signaling, then this complex formation is not allowed. This complex are no more together, they are individual proteins. In fact, this GSK will phosphorylate some other proteins here. In that case, beta catenin will be free. If this complex is not there, beta catenin will not be degraded, it will be free, and it will be able to enter the nucleus. This is the nucleus where it can bind with some other transcription factors to trigger the expression or transcription of the required genes, which in this case we are talking about large micromere cells, to specify them into mesenchymal cell, skeletogenic mesenchyme, which will be making the skeletal spicules in sea urchin. Okay, so micromeres undergo autonomous specification to become skeletogenic mesenchyme because of this transcription of the mesenchyme specifying cells, and that is because of this beta catenin signaling. And these micromeres produce initial signals that specify the other two tiers of cells also. Because these are specified now, they can specify large micromeres to the veg2. Veg2 can give veg1. So that's why they will be giving autonomous. First, they will get specified by autonomous, then they will be making by condition. Molecule responsible for specifying micromere is beta catenin, which accumulates in nuclei of those cells which are failed to become ectoderm and mesoderm. Transcription factor, this is activated by WNT pathway. The accumulation is autonomous, responsible for specifying vegetal half of the embryo. And during oogenesis, this DSH, it becomes located in the vegetal cortex of the egg. Okay, that is how in only the cells which are towards the bottom, only in those, if DSH is present, then only WNT can activate it. And then only beta catenin can enter the cells. So the first option is correct, that DSH is localized in vegetal cortex. Then also we understood that beta catenin predominantly will be present in micromeres and little bit in the veg2 tier of cells. So something like this. This is the bottom view of the sea urchin embryo. These are the micromeres, and on top of that you have the veg2 layer of cells. So orange color nucleus means that beta catenin is more concentrated here, and in the veg2 layer they are less concentrated. Okay. So there is a treatment which is done. So in the first cases, if you see over here, the first case, during normal development, beta catenin accumulates predominantly in micromeres and somewhat less in veg2 cell. So this is correct, that option C is correct. Now option D talks about lithium chloride. Lithium chloride, it actually will block the GSK. Now see, GSK was inhibited by the WNT signaling, because of which beta catenin would become free in the vegetal cells and the micromeres. But if we use lithium chloride, then not only in the vegetal and the micromeres, but in all the cells, we are injecting lithium chloride in the embryo. So all of its cells in those GSK will be blocked. That means in all the cells, beta catenin will be free. And if beta catenin free is all the cells means it will accumulate beta catenin nuclei of all blastula cells. Then wherever beta catenin is there, that cells will get specified into vegetal site. Then animal cells will not become formed at all. Okay. Even the animal cells that is towards the animal side, those cells they were supposed to form ectoderm. And vegetal cells they are supposed to form mesoderm and endoderm. Right? Now, if all the cells have beta catenin, then ectoderm won't be specified, only mesoderm and endoderm. So you can see the embryo leads to something like this. Generally, if embryo develops properly, it leads to the formation of this kind of shape, which is called as pluteus larva. Okay, but if you can see in treatment lithium chloride, it doesn't look anything like the real embryo. All right. Then cadherin expression. If you treat cadherin, it prevents beta catenin from entering nuclei. That's what the E statement said. That if you prevent the beta catenin from entering the nuclei, vegetal fates are not specified. Now, in this case, it's the other way around. Beta catenin is there in the micromeres and in the veg2 layer of cells somewhat, but they are not allowed to enter the nucleus. Now, tell me, without entering nucleus, how can the beta catenin allow the expression of the mesenchyme specifying genes? So that is why in this case, the vegetal section, what it was forming, that is endoderm and mesoderm, will not get synthesized. Endoderm and mesoderm, they will not get synthesized. Okay, they will not get specified. Only ectoderm will get specified. So all the cells become ectoderm. The entire thing becomes a ciliated ectodermal bone. So based on this only, you can see. Anyway, C was correct. Treatment of embryo with lithium chloride does not allow accumulation of beta catenin. It's not does not allow. It does allow. It allows the accumulation in nuclei of all blastula cells, because of which animal cells also become specified as endoderm and mesoderm. So that's why this is not correct, because it will allow. And when beta cat is prevented from entering, the entire embryo will not have mesoderm and endoderm, because beta catenin cannot enter nucleus. In that case, it will become a ciliated ectodermal bone. Okay. Now moving to the next question. This is regarding amphibians and in fact this is amphibian and also the birds. Okay, basically it's a combination question. They have given collar sickle some areas or tissue regions in amphibia and chick embryo. Collar sickle we will find in chicks. Hypoblast we will be finding in amphibia, means we will find in the chick only. Primitive groove we will find in the, actually all these are chicks only, but column 2 we are comparing with the amphibians. Okay, these all were tissues in chick, colocecal, primary hypoblast, primitive groove, Henson's node, and column 2 are some regions which either are describing them or they are comparing with the amphibian tissue region. Okay, so tell me which out of these options seem correct to you. This is from September 2022. So this kind of comparison questions in animals are often asked in CSIR. Okay, they will compare two different organisms, either the gastrulation process or the cleavage division or the axis formation. They will compare like this. Yes, any guesses of this? Okay, Sarita says 2 again. So first of all, we will look at the comparison. Yes, you're right. The gastrulation process of amphibian. Okay. So this is for chicks, right? What you are observing here is a top view. This is the top view, chick embryo, which is viewed from the above. Now, inside part, if you see here, this is the yolk content, because the down part, what you see, the yolk of the chick egg, it is too much. Okay, so if you have this egg, entire egg, so almost entire thing will be yolk. Okay, and the top part, this will be free, and here itself the cleavage divisions will occur, like division of this embryo, that's what will occur. That's why if you view from the top, this is the diagram viewed from the top, you are able to see these cells that has divided, and underneath you can see the yolk content. Okay, now this diagram is also like that, only it is little bit zoomed. So these all are top views. Okay, so what happens from the posterior side, the cells start moving towards the top. Okay, so they will start moving towards the top like this. All right, so when they move like this, if we cut a cross section here, okay, you can see we cut a cross section. So basically this is the entire yolk. All right, and on top of the that you can see the blastula. So now how the cells are going inside, you can see the cells are going like this, and they are coming back like this. Then again they are going like this, then they are coming back like this. That means inside. See, from the sides they are going, and then inside they are coming. From the top they are going, and from the inside they are coming. Okay, so this is how the gastrulation takes place in birds. If you can see a closer view, the posterior side cells then start moving, and a gap develops, a groove, a depression develops, through which the cells will start migrating. Follow the arrows. That is how they start moving towards the front. And slowly this depression will elongate. This depression is what is called as the line which creates, it is the primitive streak. Okay, and here you can see the line which elongates, its primitive streak, and the depression which forms, it is primitive groove. All right, and the tip of it is what is Henson's node. The cheek blastopore cells migrate inward through the primitive groove and the Henson's node. Henson's node is the tip part of the streak, and it is responsible for specifying, it acts like an organizer. Cells generated in Henson's node, they pass into the gastrula, migrate anteriorly, and they will form head structure. So basically the embryo will develop like this. Like this, it will be the anterior structures, and down will be posterior. Okay, and if you see the cells, these are the Henson's node, they will migrate anteriorly. Inside arrows you see the cells are going like this, and they are migrating towards the front. Okay, and here they are migrating towards the surface. Okay, I mean from the surface, but they are going towards the side inside. So that is how you have three layers: the top layer, which is ectoderm, then the bottom layer, which is endoderm, just on top of the yolk, and in between them you have the mesoderm cells. When you look at the amphibian embryo, from the sides you have this dorsal lip of blastopore, which starts the movement like this. The gastrulation happens. So like this, they are moving inside. Okay, the archenteron begins to form like this. They go inside, and the blastocele will be slowly displaced. You can see the endoderm and endoderm and mesoderm both are going inside, whereas the ectoderm is completely covering it. So there are various migrations which happen. Basically, this side you have the dorsal blastopore lip, okay, which is acting as the organizer. So it releases all the components, DMP inhibitors, so that this side of the embryo can get specialized as dorsal and the other side as ventral. And as you saw, the cells start moving through something called as dorsal lip of blastopore. Okay. So when we compare these two, when we take this, and yeah, Kohler sickle was actually these cells. These posterior cells, what were there, no? That started to migrate in front. So those are giving the signal for the migration of the streak. Basically, they are starting the process of streak formation, tip of which is the organizer. So they are kind of initiating the organizer. Okay. So colar sickle, they are basically the local thickening of the epiblast, which is found as the posterior edge of the area pellucida. Epiblast simply means the top layer. Whatever you're seeing the top layer, no, that is the epiblast. This layer is the epiblast. So from here only the cells go inside. Below the epiblast, one layer is that is hypoblast, but slowly the endoderm comes means hypoblast will be replaced. So the posterior end of the epiblast is what is the colar sickle. And area pellucida is the area, this yellow line you see, yellow color area. So those areas will not be transparent because here the cells, actually that is yolk anyways, but what happens is in this embryo, the central part of the cells, it's only one layer of cells. So that's why you can see inside. But the outer layer of the blastoderm, which is like a layer, blastodisc rather we say in chicks, you cannot see through it because it is inside also some cells are there. So if you draw from the sides, if this you draw from sides, it will look like this. This is the, if you draw the egg like this, okay. So this is what is your um, blastodisc, all right. So here you will have only monolayer of cells. Only monolayer of cells will be there of the epiblast. Okay. And in the sides, you will have many cells. Okay. The reason is because from the central part of the blastodisc, as a 3D only, you can imagine, I have drawn from the sides. The central part will undergo apoptosis. So cells will be killed over here. That's what generates the blastocele. Okay. So this top layer of cells, this is the area opaca, area pellucida, and then these are the area opaca because you can't see through these cells. So in the area pellucida, the last cells, group of clump of cells, that is what is the colar sickle. Okay. So that's what you can see, local thickening of the epiblast formed at posterior edge. Then primary hypoblast. So hypoblast means down. Hypo means down. The cells which are present below the epiblast. And they in fact come from the epiblast only. Okay. From this epiblast, one cells are released, some few cells. They delaminate. That means they leave this epithelium and they come out as loose cells. Later, one more layer of cells start migrating from the colar sickle and they form a layer underneath which is called as the hypoblast. So B will be 1, A will be 3. Primitive groove. It is similar to the amphibian blastopore. Groove means the depression through which the cells go inside. So it is similar to the blastopore. And Henson's node, as I said, it is acting as the organizer. So even in case of amphibian, the organizer is dorsal blastopore lip. So that's why D will be 4. Yes, option 3 is the right answer. Okay. So in case you are not aware, we Biotecnika have decided to help our students who are trying to appear for CSIR exam. Okay, so there we have launched this series which anybody can access. All you have to do is you have to just download the Biotecnika app. That is Campus app you have to download. Okay, Campus app you can download in your mobile, and from there you have to search. So recently in our YouTube channel, you can find a video how to access this T3 test series. We are giving you exclusively Part C questions. As you know, Part C weightage is more. So that is why we are giving you exclusively Part C questions. 10 questions every day you will be getting. Okay, for practice. So that's free of cost and you just have to practice it so that you people can clear. Okay. And in case you are unable to solve the questions and you feel that no, you need a proper preparation, then also we have a solution. You have the Lakshya batch, which is for both CSIR NET and GATE, and you will get all these features in this course. So all you have to do is you have to call in the numbers and or else you can directly visit stores. So this is also a similar question that we saw in June 2021. So this is also comparison between the parts of the chick and the amphibian. So now that you know what are the comparison areas, so anyone wants to attempt this now? The blastocele and blastopore lips, all these are in the amphibians. As you can see, column 1 is in amphibians and column 2 regions are in chick. So you just have to match which is having similar function or which is analogous to this. Yes. Who can say? See, just now we saw that the dorsal lip of blastopore, this one anyways you can tell. Henson's node is also the organizer, dorsal lip of blastopore is also organizer. So 3, C is 3. But C is 3 in both of them. So anyone can tell whether it is option 2 or 4 among that. Just try to remember where blastocele was present. As I told you, remember the diagram, the blastodisc. So you have the unilayer epiblast layer of cells, and slowly one cells will be released from here, like on top of the yolk. Between the epiblast and yolk, you will have the gap, that is the blastocele, isn't it? So blastocele will be present between epiblast and hypoblast. Here one more layer of hypoblast will be formed, and between that your blastocele will be there. Okay, so yes, A option will be 4. So that's it. Out of 2 and 4, if you know 2 options means your answer is already there. Yes, very good, Sarita. That's the right answer. Blastopore lip. So lip means in the amphibian embryo, you had this part, right? The depression that you had through which the gastrulation was starting. So that is what is compared to the primitive streak, because primitive streak is also a result of gastrulation. And because in amphibians, the dorsal lip of blastopore, from there only the gastrulation starts, because of which the lip creates. The cell starts migrating inside, that is why the lip is created. Okay. So B is 2. Blastopore is the actual depression through which the cells go inside. So it is compared with the primitive groove. And nucleus center, it's the one which initiates the formation of organizer. So in amphibian, if this is the organizer, nucleus center will be present below that. So it's the vegetal cells, endodermal cells, which initiate the formation of dorsal mesoderm, which is basically the organizer. So similar thing in birds or chicks, posterior marginal zones, as I told you, which is before the colar sickle. I mean, further away from the colar sickle. So they will initiate the formation of the organizer region. So nucleus center is comparable with that. In case if anyone is looking for good tools of revision, because when it comes to CSIR, you have lots to study, and that too you might get confused between this process and that process. So we have our concept revision tools, which is concept table book, axis formation in amphibian, axis formation birds. So you can see all the points regarding the organizer or the primitive streak or the BMP, which is ventralizing agents. Okay, so those all the comparison points are present in the book for all units. It's there. So many tables, whatever can be compared from all the units. So these are good books, even concept wheel also. Again, you can visit stores or you can give us a call if you want to avail these kind of revision tools since exams are near. Okay, so this is the explanation of what we just saw. Now let's look at Drosophila, little bit in Drosophila. This is from June 2018, but questions based on this topic also often they are asked. So this is regarding torpedo. It's basically the dorsal ventral axis formation in Drosophila. Torpedo is known for a receptor for gherkin. Now if we have deficiency of torpedo gene in Drosophila, it will lead to the ventralization of the embryo. If there is a deficiency in torpedo, then ventralization will be done. So what do you understand for that? that torpedo activation will be causing dorsalization. Okay. In an experiment, germ cell embryos from a wild type embryo, germ cell precursors from a wild type embryo, that means torpedo will be present. It won't be deficient. They were transplanted into embryos whose mother carried the torpedo mutation. Mother has torpedo mutation, but the embryo is wild type. Now you need to think, where is torpedo present? And another reverse experiment was done. Transplantation of germ cell precursor from torpedo mutants. This time you have taken the um, germ cell precursors which are actually torpedo mutants, and you have put them into wild type mothers. Okay. The torpedo deficient cells developed in a wild type female. So if you take the germ cells torpedo mutant, that is the second case, and you have put it into a wild type mother, it showed normal dorsal ventral axis. But in the first case, where you taken the wild type germ cells and you have developed in a torpedo deficient female, in that case you can see that there is no normal dorsal ventral, but it shows ventralized egg. So what does that mean? The the female, that is the mother, if it is not having torpedo, then the dorsal ventral axis is getting affected. Which means, if you remember the structure of the egg, Drosophila egg, so you have this egg present over here, and surrounding you will have this follicle cells. Okay, this is the Drosophila egg, and surrounding it will be the follicle cells. And here will be some more cells which are called as the nurse cells. And this is what releases some of the components. So basically this follicle cells will be having this torpedo receptor. And follicle cells are part of mother, even nerve cells. This entire thing is the mother. Only the egg is what is form the embryo, what will form the embryo. Okay. So this egg will release the signal which is called as gherkin, and the receptor is what is the torpedo, which should be present in the mother. So following conclusions were made. Zygotic contribution of torpedo is essential. No, in that case you wouldn't have got this kind of expression. Clearly the maternal contribution is essential. So B is right. Okay, so if B is right, means either 1 is right or 2 is or 3 is right. So anyways, in both C is common, which means C is correct. Since torpedo is a receptor for gherkin and the follicle cells around the part of the oocyte where gherkin is expressed. Now generally, if you would know, in case of Drosophila, before the dorsal ventral axis formation, the nucleus moves towards the future dorsal side. Whichever side dorsal side has to be done, the nucleus moves towards there. And wherever this nucleus is there, they will release the signal gherkin. Okay, they will release the signal gherkin. Receiving that signal, the torpedo receptors, what are there, they will get activated signaling response. And because of that, pipe protein will not be synthesized towards the future dorsal side. But towards the future ventral, remember this entire thing is the egg. Okay, this part is the egg. Now in the ventral side, there is no gherkin because nucleus is on the other side. So torpedo will not get activated. Receptors are present this side also, follicle cell, but they don't get signal. Okay, that is why this side, future ventral side, pipe protein will be synthesized. Now pipe protein has a big role in giving some signaling responses, because of which ventralization happens. If pipe protein is not there, dorsalization will take place. Okay, so it is likely that torpedo is expressed in follicle cells. That's true. Gherkin signaling initially dorsalizes the follicle cells, which in turn sends signal to organize the dorsal ventral oocyte. So gherkin signaling, as I told you, it will be towards the dorsal follicle cells, and they will send the signal. Because of that torpedo signaling, they will signal the dorsal ventral polarity. So yes, D is also right. So you have got your correct answer. That is B, C and D. Option 1 is right over here. Yes, that's right. Very good, Sarita. Now let's see why E is wrong. Gherkin signaling initially dorsalizes nerve cells. No, nerve cells, these are the nerve cells. They don't get any signal. The torpedo is only for the follicle cells, which help in generation of the dorsal ventral polarity in the oocyte. Okay. So see, this is the signaling which I was telling you. The yellow colored thing is the Drosophila egg. Okay. And surrounding it, you can see all the follicle cells are there. These are your nerve cells, ovarian nerve cells. So anyways, the oocyte is later going to form the embryo after fusion with sperm, right? So the nucleus right now is towards the dorsal side follicle cells. If this part is zoomed, you can see the nucleus moves towards the dorsal side where they will bind to the torpedo receptors on the follicle cells towards the dorsal side, future dorsal side. And here they will inhibit the pipe protein synthesis. Towards the ventral side, pipe protein will be there. Now, why pipe protein is so important? Now remember, the embryo, egg, when it forms the embryo, in Drosophila embryo, there is no cell division in the initial stages. Only the nuclear division takes place. So you can see these are the nuclear divisions. These are all nucleuses, which will be subjected to a morphogen gradient so that the anterior posterior areas are specified properly. Okay. So this side you have the nucleus. In this case, cytoplasm has a molecule. The embryo of cytoplasm, they have a molecule which is called as dorsal. It is expressed, okay, but dorsal is generally bonded by cactus. All right. Now this side the follicle cells are there, which have already got the signal of ventral by the pipe, means by not receiving the Gherkin signaling, right? They have the pipe protein synthesis. So they are destined to form the ventral. This side pipe protein is present. Okay. And towards the future dorsal side, pipe protein is not there. No pipe. Okay. Because of which there is some signaling so that it will form dorsal. So what exactly is this signaling? See, this dorsal is bonded by cactus. Dorsal is bonded by cactus and it is present all over the cytoplasm. But towards the ventral side, the follicle cells release the pipe, which gives some kind of signaling. I'm telling you what signaling is done, because of which this cactus is degraded. If pipe protein is there, because of this entire signaling, ultimately the cactus will be degraded in the ventral side. Okay, so dorsal cactus, those cactus will be degraded in the ventral side and the dorsals will be free to enter the nucleus in the ventral side. Okay, so if dorsal enters the nucleus, you might think it will lead to dorsalization of this side, but no, it's the other way around. Generally, genes are named by their mutational phenotype. So wherever the dorsal will enter the nucleus, whichever side, that is towards this ventral side, it will cause ventralization. It will activate gene expression of the ventralizing cells. Means ventralizing genes it will activate. Okay. Then why it is called as dorsal? It is on the mutational phenotype. If dorsal is not there, then it cannot enter the nucleus and it cannot activate the ventralizing cells. So in that case, automatically the cells will specify dorsal. So if dorsal is not there, dorsal protein is not there, then the cells get specified as dorsal cell types. That's why it's named as dorsal. But always remember this summary, that dorsal causes ventralization. Okay. So what is the entire signal? And okay, meanwhile, what is happening at the dorsal side? There is no pipe protein in the dorsal side, because of which the cactus will not get degraded. So here towards the future dorsal side, dorsal is not free. They are bonded by cactus towards the dorsal side. So they cannot enter the nucleus in the embryo. So if dorsal cannot enter the nucleus at this side, then no ventralization. That is why these nucleus get specified as dorsal cells. Okay. So the signaling process, you remember, the pipe protein along with some other X protein, they will do a series of proteolytic cleavages. That means they will cleave GD, a part of GD will cleave snake, that part will cleave ester, that part of ester will cleave spazole, and that part of cleaved spazole will go and bind with the receptor toll, okay, which is present on the egg membrane. And toll will activate something called as PEL and tube. So these things you need to know. Pell and tube are actually acting as kinases. So they get activated on the future ventral side because of pipe protein signaling. If these kinases are active, then the cactus is phosphorylated. And if cactus is phosphorylated, then the dorsal protein will be, means phosphorylation means ubiquitination will happen. So cactus will be degraded in the ventral side and dorsal can enter the nucleus in the ventral side. But in the dorsal side, okay, so here we have here we have this dorsal sulfate which is present. Okay, there is no pipe protein signaling. Because of that, the pipe protein signaling is not there. That is why towards the dorsal side, the cactus will not be degraded. Okay, because Pell tube in this side will not be activated. And if cactus is not degraded, then dorsal will not be free and they cannot enter the nucleus in the dorsal side. So automatically they will cause dorsalization. Ventralization cannot be done. So these are the events which I just explained here. So this was a question similar subject in December 2017. So now that you understood the concept, can anyone try answering this? No guesses? See, these are here they are asking about. Okay, Sarita says option 3. Let's say which combination of the above events will occur in the presumptive dorsal side of the embryo which is deficient in maternal gherkin. So this is a mutational question. That means the embryo gherkin signaling is not there. Now remember, if gherkin signaling is not there, okay, then the follicle cells which are there, the gherkin signaling will the gherkin signaling will activate the follicle cells generally in the dorsal side. But if gherkin is not there, then it will be same as ventral side, because in anyways in ventral side you don't get gherkin. So basically what is supposed to happen at the ventral side will be resembled in the dorsal side if the embryo doesn't have gherkin. So what all things takes place in the ventral side, now you remember? Torpedo, does it get activated? No. Torpedo gets activated only in the dorsal side because of gherkin. Gherkin is not there means no torpedo activation. Pipe protein, does it get synthesized? Yes, in the ventral side and now even in the dorsal side because there is no torpedo activation. So pipe protein will get synthesized because torpedo activation leads to inhibition of pipe protein synthesis. So since there is no torpedo activation in the future dorsal side now, that is why pipe protein will be synthesized even in the dorsal side. Pipe protein means cascade of protease activity will also be there. One by one, each of the molecules will be cleaved. Okay. Because of which the Pell tube will be activated. And because of that, cactus will be phosphorylated, not dephosphorylated. So D is not correct. It's written dephosphorylated. Okay. Because of those activity, cactus will be phosphorylated in the dorsal side so that it can be degraded. Then only dorsal will be free to enter the nucleus. Right. So D is not correct. E is again correct because cactus is phosphorylated. It will be degraded. So dorsal will be free. It will enter the nucleus of syncytial blastoderm stage embryo. So that's why here the correct answer will be B, C and E only. Yes, Sarita, that's right. Very good. Now another question from Drosophila. So this is regarding the anterior posterior compartment. Basically, you would be knowing the segment polarity genes. So this question is regarding that. The anterior posterior segment compartment of each segment of Drosophila is defined by wingless and engrailed genes. The following statements are given towards how they are regulated. As you know, the Drosophila signaling, it starts with the bicoid and nanos, which are the anterior and posterior determinants, morphogens respectively. Bicoid is for anterior and nanos is for posterior. And these are the maternal effect genes because they are synthesized from the egg itself as mRNA. When the embryo gets fertilized, they will get translated as proteins and they form a gradient. Accordingly, whichever nucleus is getting high concentration, they will become head. Which are getting intermediate, they will become thorax. And whichever side the nanos is there, they will become the abdomen area. Now, accordingly, these nucleuses, they will activate some few further genes which will specify the area. So those are zygotic genes. So immediately one of the zygotic genes are gap genes. So they will create broader segments in the insect embryo. You know, insects have the segmentation in the body. Gap genes will activate pair rule genes, which further create some wider some narrow segments. So some more expressions will be done. Total 14 segments are there. And then each of these segments, they will get anterior posterior polarity so that they can accordingly get specified which side will be anterior, which side will be posterior. So those only which are called as segmentation genes, wingless, engrailed, all these are involved in that. So few statements are given regarding this, how the anterior posterior compartment of segments are done, rather how they are regulated. So you have to select which is the correct answer. Yes. Any guesses here? What will be the correct answer? Any guesses? Here also we have signaling is associated with this question. We have the sonic hedgehog signaling, we have the WNT signaling. You can see wingless is the present, right? And it's just that some names will be different. So we have this hedgehog signaling. There we have something called as C and glee protein. If you know those signaling processes, you will know. So cubitus interruptus is the C protein. So where it is expressed. Okay, let's see, Sarita. So see, this is what is the signaling regulation happens. Basically, these are all the 14 segments. As you know, Drosophila will be having segment and parasegment also. Parasegment is basically the posterior part of the previous segment and the anterior part of the next segment. Okay, so different, these are all the pair rule genes which are activated. Anyways, I'm not going to detail of that. But here one such segment is shown. This entire thing is one segment. Okay. Now you can see in this segment zoomed and shown in the posterior part of the segment, engrailed, the cells which are there, they will be expressing engrailed, and the cells which are just anterior to the engrailed expressing cells, they will express this wingless. Okay. So that two cells are shown over here. This is the wingless expressing cell and this is the engrailed expressing cells. That means these posterior part of the segment cells will release engrailed. Okay, they will be releasing this transcription of engrailed, and along with that they will release this diffusible signal called as hedgehog. Now hedgehog will come to the cells anterior to it, that is these cells. They will receive the maximum of it. Okay, because these cells only will have the receptors for it. These are the patched receptors. When hedgehog binds to these patched receptors, they will activate a protein called as smoothend, and smoothend will cleave this protein called as cubitus interruptus. That cleaved part of the C protein, which is also glee protein in some species, that cleaved part acts as a transcription factor. Because of the signaling, it gets cleaved and it goes to the nucleus and transcribes wingless. So wingless is released from here and again it goes to the same cells. So it is kind of a reciprocal signaling. They maintain each other. Okay. So these side cells cannot get engrailed signaling because they don't have the receptor for it. This cells, which is the posterior part of the segment, Only they have the receptor, which are the frizzled receptor. So that is why only they are expressing hedgehog. And since hedgehog is released from these cells, and this cell is the closest to it, so that's why in this cell wingless will be released. This is how they maintain each other. Now see, wherever this engrailed is expressed, it is the posterior of a segment, correct? But it is also the anterior of a parasegment. So engrailed is expressed in posterior of a segment and in anterior of the parasegment, if you take the parasegment part. Okay? Whereas wingless is expressed in the posterior of the parasegment. So this is the parasegment. So wingless WNT, when it binds frizzled receptor, these all you know, it activates DSH, disheveled. Disheveled inhibits the GSK complex. So ZW3 is the analog name in Drosophila for GSK. And because GSK is inhibited, the armadillo is free, which in this case is beta catenin, which can enter the nucleus and activate the expression of engrailed and hedgehog. Right? So you can see here, yes, Sarita, your answer is right. Wingless is indeed a secretory factor. That's why it is going to the nearby cell and binding to it because of signaling, it is able to express the engrailed. Engrailed is secretory factor. No, engrailed is not secretory. Hedgehog is a secretory factor. Engrailed is transcribed, but hedgehog is releasing the signal and forms a long range concentration gradient. No, it doesn't. Engrailed regulates wingless. So that's what I said. Engrailed will be activated, but it actually regulates the wingless in the cells anterior to them through hedgehog. And it doesn't form a long range, it is forming only a short range. That is why the cells only just next to them are getting the hedgehog. The other cells are not getting it. So it has a short concentration gradient. C is correct. Beta catenin homolog is the signaling molecule which is upstream of engrailed. Upstream means you can see um, the sequence wherever it is getting engrailed. Upstream of that is this armadillo, which is nothing but beta catenin. And beta catenin is made free by GSK. Okay, so beta catenin homolog, which is armadillo, it's present upstream of engrailed, which gets cleaved by GSK3 homolog, which is ZW3 in this case. That is correct. But if there is signaling, WNT signaling, then this will not take place. Okay, but anyways, this is correct statement. C is an intracellular signaling protein in the engrailed expressing cells. No, not at all. Just now we saw C is expressed in the wingless secreting cells. Okay. Engrailed secreting cells means WNT signaling. And wingless secreting cells means the hedgehog signaling will be taking place. So C will be acting in the wingless secreting protein, not in engrailed one. So that's why E is wrong. A, C and D, option 4 is the right statement. Clear everyone? Okay. So that's all from my end. Thank you. I want to remind you about the T3 series. Don't miss it. And also if you want to be a part of the lecture batch, then do not delay anymore. Thank you. And next class, we will be talking next week. One more Unit 5 session will be there. We will discuss Unit 5C, which is the organogenesis. Thank you.